Hello everyone. Today I'll be presenting Hassabis' research on protein structure prediction using AI, the work that was recognized by the 2024 Nobel Prize in Chemistry. The 2024 Nobel Prize in Chemistry was awarded for two revolutionary achievements: computational protein design and protein structure prediction. Professor David Baker pioneered computational protein design, while Demis Hassabis and John Jumper was recognized for their work on protein structure prediction. Today's presentation will focus on their achievements, the AI model, AlphaFold. First, what is the protein folding problem? This relates to the central paradigm of biology. Every protein starts as a first-dimensional sequence of amino acids, but it falls into a unique 3D structure. And it is 3D structure that determines the protein's unique biological function. The grand challenge for 50 years was to predict this 3D structure from the sequence alone. But how difficult is this problem? It is best explained by Levental's paradox. In the 1960s, Cyrus Leventhal hypothesized that if a protein tried every possible conformation randomly, it would take longer than the age of the universe, about 13.8 billion years, to find the right one. But in nature, proteins fold perfectly in just a few seconds. This contradiction is the core of the problem. How does a protein find the single correct structure so quickly without trying all possibilities? It implies that folding is not random, but follows specific pathways and physical chemical rules. Finding these hidden rules has been the goal for the last 50 years. So, before AI, how do scientists determine these structures? We rely on experimental methods. The most famous is X-ray crystallography, which involves crystallizing a protein to determine its high-resolution 3D structure. There is also a NMR spectroscopy, which analyzes proteins in a solution to get structural information. And more recently, cryo-EM, which visualizes the structure of very large protein complexes by flash freezing them. Now, this is the most crucial point. The 170K protein structures collected over decades from these experiments became the textbook. It means the training data that the AI deep learning model AlphaFold learned from. AlphaFold's principle works like this. First, it performs a multiple sequence alignment, or MSA. It compares a specific protein across many species, like humans, mice, and chimpanzees, to find residues that are conserved. It means that they are evolutionarily important. It means co-evolutionary patterns from these alignments to predict which amino acids will be close together in the 3D structure, even if it is far apart in the sequence. Finally, it combines this information with physical constraints to build the most realistic 3D structure. But before AlphaFold, these experimental methods created a critical problem: the sequence structure gap. Think about this. Thanks to NGS sequencing technology, we have sequence data for tens of billions of proteins. But how many 3D structures did we have? It is only about 180K. That's not 10%, it is less than 0.1%. This massive gap was the single biggest bottleneck in modern biology. We have the list of all the sequences, but we have no structures to understand how they actually worked. So, why this bottleneck happened? It happened because the experimental methods themselves have fundamental limitations. For X-ray crystallography, the biggest challenge isn't the X-ray, it's the crystal. You must purify a protein and force it to grow into a perfect, solid crystal. This process alone is an art form that can make tens, months to years. And for many critical proteins, like membrane proteins, which are the targets for most drugs, crystallization is most often impossible. For NMR spectroscopy, the limitation is protein size. It works well for small proteins, but many of the complex machines in our cells are simply too large for animal to analyze. Here, AlphaFold changed everything. Let's directly compare the old way versus the new way. Look at the time. Months to years becomes minutes. The cost, the need for very high cost equipment is replaced by low cost computing. And most importantly, the success rate. A difficult, low success experimental process is replaced by a prediction with accuracy close to experimental results. The key point is this: AlphaFold didn't just improve the old methods, it overcame the fundamental limitation of cost, time, and physical constraints that created the bottleneck in the first place. The impact of AlphaFold on modern biology has been immense. First, it released the AlphaFold-DB, 200 million plus predicted structures. While humanity saw 180K protein structures in 50 years, AlphaFold, the AI model, solved the 50-year sequence structure gap only in 1 year, making over 200 million structures public and accelerating global research. It also helped respond to infectious disease. AlphaFold enabled the rapid prediction of SARS-CoV-2 protein structures and revealed how mutations like Omicron affect infectivity and immune evasion. And it aids in research for neglected disease. It is used by the DNDi to predict parasite protein structures for Chagas disease and leishmaniasis, accelerating drug discovery. But AlphaFold isn't just an answer. It is a revolutionary tool for all of biology. For cancer research, it can instantly predict the structural changes in mutant proteins like P53. This opens a new path to designing targeted anti-cancer drugs. For apoptosis research, it helps us understand the fundamental process of life at a molecular level by predicting how signaling proteins interact and bind together. In summary, AlphaFold has succeeded in predicting the 3D structure of proteins from only their amino acid sequence with higher accuracy. Thank you for listening.